Hello everyone. Today we will discuss about the instrumentation about on the fluorescence spectroscopy. We'll try to look at the details of the instrumentation. When you go for the instrumentation based on the fluorescence, fluorimeters are there. Fluorimeters are spectroscopy oriented instruments. You have microscopes, which are biology imaging based equipments, high throughput plate readers, imaging systems, intravital imaging systems. They are all biology based instruments. We'll try to understand the spectrofluorimeters in detail in this session. When you look at the spectrofluorimeters, there are different kind of nomenclatures they use it. The spectrofluorimeters, they name it like fluorescence spectrophotometer, fluorimeters, fluorometers, spectrofluorimeters, spectrofluorometers, luminescence spectrometers. They are all referring to the same equipment, fluorescence spectrophotometer. Otherwise, you call them as a luminescence spectrometer. Luminescence always refers to here photoluminescence, which includes fluorescence and phosphorescence. So we should not confuse with this nomenclature. Maybe it looks like they are dealing with different machines, but they are all referring to the same equipment, fluorescence spectrophotometer. Now, when you go for a fluorescence spectrophotometer, there are a number of manufacturers worldwide involved in the manufacturing and selling of the equipments. One of them is ISS, Fluoromax or Job Envion, Japanese-based company. They deal with the spectrofluorimeters, lifetime spectrometers, and so on. And Quantum Master from UK and Canada, they also deal with the spectrofluorimeters. Now, in this picture, what you see is the spectrofluorimeter that we were using in our lab, which was bought in 1978. Even now, it still works, which is a mode of manual machine, where you can see all the knobs which can really change the slit weights, wavelengths of the monochromators, excitation, emission, scan speed, selection of monochromators, all that you can visually see it. And we connected this equipment to the computer at the later stage. We use this equipment for the MSc practical purposes, where students can understand what is happening on the machine when you change all these knobs, physically see it. Now, if you see the latest machine, which is from a Job Envion Fluoromax, you don't see any knobs on top of it. And all of them, all the controls, manipulations, change of settings, everything is done through the computer, but you don't physically see it. That's a kind of a feeling difference you will see between the old machine and the new machine. But anyhow, it has got the advanced features which can do a lot of advanced experiments, and compared to the old machine, which was basically a normal analog machine which has a digitized output. Digitized output is connected to the computer, whereas this one is a photon counting machine, which is a TTL, which is more sensitive when compared to the other machine. And if you look at the what is there inside the spectrofluorimeter, if you look at the block diagram, one can find out that there is a light source. You have the monochromator, which is excitation monochromator. This is used to select the wavelength of the excitation. And you have the polarizer, which is an excitation polarizer, sample compartment. and we measure the fluorescence intensity from the sample at 90 degrees geometry. And the choice of the 90 degrees geometry is to avoid the intensity of a transmitted light interfering with the measurement of fluorescence. Fluorescence intensity will be coming after the absorption of photon in all directions, so any directions you can use it. And in order to avoid the interference from the excitation light or transmitted light, and preferred geometry of measurement of fluorescence intensity is 90 degrees geometry. And where they place the emission polarizers. If you are interested in measuring the anisotropy behavior of the fluorescence, you have two polarizer. One is the excitation polarizer, other is the emission polarizer, and there is emission monochromator. This is to really distribute the fluorescence intensity over the wavelength to record the fluorescence, and then this is signal is sent to the detector, and finally it is recorded in the computer. This is how you see the a whole picture of the fluorimeters. Now if you want to understand what is inside, you can also look at it. And there also another type of fluorimeters, a kind of a photoluminescence setup. They use it in the material science applications. This is based on the laser excitation, and if you really want to have a UV excitation, they really opt for the laser at the source, and make the optical systems in between. You have the light falling on the detector and collection optics, and then fiber optic coupled to the spectrometer, finally to the computer and analyze it. So this is generally used for UV or low emission intensity applications, or to understand the bandgap materials, and mostly solid state applications you will see it. And here is a spectrum that I have given you as a case study: gallium nitride in oozite structure, and emission is at 365 nanometer, and excitation is 266 nanometer laser. And this is also another kind of a setup, whereas in the previous block diagram which I shown you, it's only a lamp based excitation. It's a normal spectroscopy application you will use it, whereas this is a more of material science application. Both of them measure the photoluminescence only, but here you have a wavelength limitation. There is a spectral distribution of wavelength, but the intensity of the source that you are using in the previous setup is lower when compared to this. These are the, each one has got their own applications and advantages. Now if you go to the details of the block diagram which I have shown you, what you see on the boxes, basically light source. It's a xenon lamp. They use it in a spectrofluorimeter, and it is sent into the monochromator, which is excitation monochromator. In the excitation monochromator, three optical components you will see it. The focusing optics on the grating. Grating is a reflecting grating, and then another optics and sends out the light to the sample, and the emission from the sample is collected again, passed through the emission monochromator, and then detector is detecting the signals. So finally we record an emission spectrum. These are the details of the components inside. We will look at the details of each one of the components, starting from the light source to the detector, and what kind of a sample compartment, what kind of a monochromators they use it to acquire the fluorescence and the other intensity data in this spectrofluorimeter. Now, in the meantime, we should also look at there are different types of the luminescence we already discussed in the basic session, which is photoluminescence. And photoluminescence comprises of a fluorescence, which is called as a prompt fluorescence if the relaxation is from S1 to S0, and delayed fluorescence is basically S1 to T1, and then T1 to S1, and then S0. It is again called a delayed fluorescence. And phosphorescence, T1 to S0 is a phosphorescence. So you can measure all these luminescence behavior in a spectrofluorimeter. And you also have other luminescence behaviors like chemiluminescence, bioluminescence, triboluminescence, cathodoluminescence, thermoluminescence, electroluminescence, and so on. All these cannot be measured. It is only the photoluminescence properties can be measured in this spectrometer. Now, if you go to the details of the individual components, let us start with the light source and then go to the detectors, wavelength selections, and polarizers. Now, before going into the light source, we have already seen it. Light source is important requirement to excite the molecules, and subsequent to the absorption, the electron goes to the excited state in the femtosecond time scale, and then the electron relaxes from the upper electronic states to the first excited state, S1 state. From that state onwards, it starts emitting light, and you see the fluorescence. This is the phenomenon. This fluorescence happens in nanoseconds time scale. In addition to this, there is a non-radiative process which is also competing along with the fluorescence. Now, to do this light excitation, they use a different kind of a light sources here, and in a spectrofluorimeter, generally xenon arc lamp is being used. Xenon arc lamp as the spectral distribution from a UV to visible region, IR radiation also, and you can select the wavelength from this light source. There are ozone-free xenon lamps also available. In addition to this xenon lamps, you can use high pressure mercury lamps, mercury xenon arc lamps, and mercury xenon arc lamps gives a more intensity in the ultraviolet region. You can also use a tungsten lamps where it has an option only in the visible region. You can LEDs, you have a choice of LEDs and have different wavelengths to excite, or white LEDs, UV LEDs are also available. So there are different kind of sources. One can opt for the source as the excitation source in a spectrofluorimeter, but the most preferred one is a xenon arc lamp with the different wattages. But standard it starts from 150 watt, it goes up to 1000 watts. But spectrofluorimeters always have a choice of 150 watt up to 500 watt. You can see the fluorimeters using the xenon lamps. And this is the spectral distribution of a mercury xenon arc lamp, which has a more intensity in the ultraviolet region when compared to the xenon lamp. You can see it. Now, as I told you, the LEDs can also be used. They have a single wavelength LEDs available. Choice of LEDs one can have it to excite the molecules, and they have a wavelength range starting from 350 to 2300 nanometer. You also have the near UV LEDs available. So, depending upon the requirement and based on the analysis we want to make it, you can have a choice of LED sources. And laser sources can also be used for the photoluminescence, which is used for a material science application. So PL setup, we already mentioned about it. You can have a choice of lasers for this. You can have either a visible lines like a 532, 543, or you can have a UV lines, ECAD lasers, helium cadmium lasers, argon ion lasers, NDOG lasers, green helium neon, orange helium neon, red lanes, and Tie-Shaffer lasers. So like this, you have a choice of laser sources to excite the materials, understand the photoluminescence behavior. Laser diodes also, they can also be used as an excitation sources. The second component is the detector. When you go for detector, there are two basic family of detector photomultiplier tubes. Photomultiplier tubes are the generally used detectors in a spectrofluorimeters. Now the compact fluorimeters are available with the CCD and diode array detectors. But PMTs are very standard. They can be used for a intense light application. CCDs have a limitation. They cannot be used for intense light applications. They can be used only for a low light applications. Now in this PMT, you have one of the on the left side, which I am pointing it, is called as a side-on type PMT. The other one is called as head-on type PMT. Why we call it as a side-on type PMT is that the photocathode, which is the photoactive element or detector elements present on the side of the detector, is called as a side-on type PMT. And here it is present in the head of this. So it is the difference between the Side-on type is like having the sensor on the head of the photomultiplier tube, the head-on type. So head-on type is used for low photon applications where you want to collect large number. Collection area is larger when compared to this. So it is very sensitive when compared to side-on type. And not only the spectral sensitivity of the photomultiplier tube have to be considered when you select the detectors. Time resolution is also important. If you are going for a time measurement of the fluorescence, then you go for a short resolution, short rise time detectors. This is one of the short rise time. What you see is called as a MCP PMT, and that is the one used in TCSPC extensively. You also have the other detectors little longer than MCP PMT APDs, which can be used for a time resolved spectrofluorimeters or on FCS spectrometers. Now, these are the basic detectors. They use it in the spectrofluorimeter, PMT, MCP, PMT, APDs. Now, if you want to understand how exactly photomultiplier tube works, and this is something like a tube which has the voltage dividers, and you have the photocathode surface. When the light falls on it, you have the ejection of electron. This electron, one photon makes the one electron, and then you get the electrons get amplified because of the voltage divider circuit. Finally, you get an amplified signal. This is how the dynode chain of the assembly of the is present inside the photomultiplier tube, which really amplifies the signal, which can be used for the photon measurements, which are much sensitive than the other detectors. Photomultiplier tubes are most preferred. They can operate in the voltages of 1000 to 3000 volts. Generally, spectrofluorimeters, they have a voltage of around 1000 volts. They use it, and you have a very good amplification of the signals. This is how the classic PMT design looks like. These photomultiplier tube also have the spectral sensitivity, a different type of a family of the photomultiplier tubes. This is basically side-on type PMT, where they have the different photocathodes. Photocathodes are mostly alkali metals, so as they change the alkali metals or photocathode material, spectral sensitivity changes. They have a broad sensitivity for the UV and visible, or there are blue sensitive PMTs or red sensitive PMTs. And a spectrofluorimeter generally a broad PMT like R928 from Hamamatsu PMTs are used, and this Hamamatsu is a Japanese company which makes the photomultiplier tubes for the application towards spectrophotometers or spectrofluorimeters. And depending upon our wavelength of the emission, we have to have a choice of the fluorimeter PMTs. So this is how you can select a PMT also, and you can buy a blue sensitive, red sensitive, or a broad sensitive PMT. Now you also have the detectors in the photon counting mode. Normally PMTs can be used in the analog detection or in the photon counting mode. And analog simple, take the output from the PMT, digitize it and use it, whereas here is a power supply changes and digitizes and measure it in the form of TTL signals, and that really counts the photons. They have an advantage over the normal analog detectors. Sensitivity is very high here, and also the stability of the measurement is quite high when compared to the analog machine. Analog machine have the primary advantage of broad dynamic range, adjustable range is possible. So voltage can be adjusted. Here is a constant voltage, here is a variable voltage that you can use it. And this is the difference between both of them. But the photon counting machines are much sensitive and very good signal to noise ratio. And this is the reason why they prefer to have you can get prefer to have these detectors in most of the spectrofluorimeters. They can even collect a very good weak Raman signals in spectrofluorimeters. Now, when you go for a wavelength selection, So you have different components available. You can use an optical filters, tunable optical filters, monochromators, and spectrofluorimeter generally come with the monochromators, which are using a gratings, which are reflection based gratings. And you would have studied transmission grating and reflection grating in your physics classes. Basically, reflection grating is used for the spectrofluorimeters, and rotation of this reflection grating facilitates the selection of wavelength, and this is how they have a choice of the wavelength by movement of the reflection grating. And now you can see on the right side you have an entrance slit of the light, and there is a focusing concave mirror on the reflection grating, and then from the reflection grating you can have another concave mirror and exit slit. And now as you rotate the reflection grating, the angle changes, then you can see the wavelength also changes. This is how the selection of the wavelength is possible in the monochromators by combining slits and the rotation of the reflection grating. This is how the choice of the wavelength is happening from the dispersion of white light from the reflection grating. And once this wavelength is selected, and then you can really excite for excitation part. For emission side, you get the distribution of the light from the emission also. You can also have the other choice as a component in monochromator is a prism based monochromator is also possible. And but they have a lower resolution when compared to the grating based machines. Grating resolution, or the resolution of a grating, is based on the number of lines that you draw on the grating. They are going to decide the spectral resolution. You know about Bragg's law. N lambda is 2d sine theta. The lambda spectral resolution is decided by the line spacing drawn on the reflection grating, and that is how the reflection grating based monochromator is having a better resolution. You can even go up to 0.2 nanometer resolution, or 0.1 nanometer resolution is possible in the grating based machines. But you can have the prism monochromator also in principle, but the preferred one is the grating based. And if you don't have the monochromators, you can even have a choice of the filters. So you can have a filter channel here, insert the filter inside, and then use your selection of wavelength also. That is also possible if you are going for a filter. So there are a number of filters available, and you have a long pass optical filters, UV filters, and little bit on the visible blue filters and red filters and so on. The color you see it, and the range of the filtering action that you see on the spectrum that you are showing here. And you can select your filters based on the wavelength that you want to use it for the excitation. Now, in addition to these broad filters, you can also use broad band pass filters or an interference filters, or you can use other filters like a neutral density filters. They are basically called as attenuators. They really use it to reduce the intensity of the light falling on the detector, or intensity of light that is being collected. So these are the family of filters you should be familiar with when you are using a spectrofluorimeter: band pass filters, interference filters, neutral density filters, and broad filters or cutoff filters. Now, when you go for the monochromator, what kind of a design they try to have it is called as a Zerny-Turner design. You have an exit and entrance port, and you have the two mirrors, concave mirrors, and you have a moving z-axis rotation diffraction grating. So it can be planar or concave depending upon the geometry. So you have the slit width, and generally slit width is defined in terms of the millimeter, and the band pass you see this is in terms of full width of maximum. and dispersion is the factor to convert the slit width into a bandpass. This is what is called as the dispersion factor. Normally, if you look at the Zerny Turner design of this kind of a small monochromators, it is something like a 1 millimeter would correspond to 5 nanometer is the band pass that you get it. Then this is the way you can really have the selection of monochromators. And this is to illustrate what is inside the monochromator. So physically what you're seeing into the monochromator. Now you have the a grating which is mounted on the rotating axis, and then one is the mirror which is sending the white light and falling on this, dispersing it, and you have another reflecting mirror and sending it out. So you can really see that, and this is basically a geometry in which the optical elements are mounted, and also the monochromator components are really clearly illustrated here. And three basic components, two mirrors and one grating, can be concave and planar, or this can be planar, this can be concave, depending upon the application. They have a choice of the optical components. Now the other thing that you should also look at is the slit width. Slit width is important component which is connected with the spectral resolution. Smaller the slit width, better the spectral resolution. Wider the slit width, higher the sensitivity. So when you keep a wider slit, you get more light, sensitivity is better. When you keep a narrow slit width, sensitivity is smaller, but the spectral resolution is higher. This is how you can see that it's a trade-off, and you have to compromise on this, what exactly you want it. This is the picture telling you about a larger broad slit width gives you a broad spectrum and more intensity. And as I narrowed down the slit width, the intensity also reduces, as well as it also increases the spectral resolution. This is a very good illustration how exactly slit width. If you are interested in finding out the very good spectral resolution in your fluorescence spectrum, it's good to have the narrow slit width rather than a wide slit width. If you want to measure higher sensitivity, if you are not interested in looking at the spectral resolution, go for the a broader slit width. This is how you have a choice of the slit width to decide between the sensitivity and the spectral resolution. This is again illustrated here how exactly the band pass is deciding the sensitivity and the spectral resolution. Now you can see the 2.125 and 17 nanometer. So this is basically a band pass. And with this band pass, if you normalize it and see it, and how exactly the bandwidth, full width of maximum changes. So this is how you can see that relationship between the slit width and the band pass. Now you also should look at some of the problems that you face in monochromators. First problem is a stray light problem, and which is very critical when you are looking at the fluorescence. Stray light is generally light that passes through the monochromator besides the desired wavelength. In addition to what wavelength you will get a stray light, which will interfere with your signal. In particular, when the signal of your fluorescence is very low, and if you're measuring on the solid state materials, and you will see enormous amount of the scattering, and stray light scattering will interfere with your fluorescence signal. You have to be careful. And you also have a solution for this. So best way to avoid this is use a double monochromator to avoid the stray light. The second point that you should look at is a scattering. Is a second order scattering. Second order scattering. Sometimes people misunderstand as if it's a new emission signal coming up. But a lot of papers published as a new emission, they observe it. But it's not a new emission. It's basically a scattering or Rayleigh scattering of the gratings that you use it, and you will have a first order scattering which will be more intense, second order scattering will be less intense, but you will see exactly at double the wavelength. If you are exciting at 300, you will see the first order scattering and the second order scattering at 600. So if you are measuring the emission intensity all through the wavelength, then you will always see the interference. So you have to really remove it by putting a filter for it. It's a band pass filter. You have to use it. Otherwise, you have to restrict the recording of the fluorescence within these second order limits. And the third point you should also look at, if you want to really get a more light output, you should opt for a high throughput monochromator. So you have these low throughput problems, second order scattering, which can be really blocked by using appropriate filters, stray light, which can be avoided if you are measuring a low fluorescence signal samples by going for a double monochromator spectrometers. These are the problems you can avoid it when you are recording a fluorescence spectrum. Now, if you sometimes, after a long usage of the fluorimeters, you lose the calibration because of the mechanical movement problems. You lose the calibration. You have to recalibrate it. But some of the machines have the recalibration automatically in the spectrometers. If you don't have one, you can also do it by using a low pressure mercury lamp or using a laser. So low pressure mercury lamp, you can see that they are available and they give a UV light and visible light. You can use these visible lines and for calibrating the monochromatic readings. You can put these light sources in place of xenon lamp and get the light out of the excitation monochromator and put a scatterer in the sample compartment and then pass it through your emission monochromator and see these two are in same line, and that is how they calibrate it. There are two types of mercury lamps they use it. You have a pen ray lamp and or you have a cylindrical housing type of lamps. You can use it to see the spectral distribution of these lamps. You can see that I given you a emission lines from these mercury lamps. Strongest one is 254 and you have the other lines 365 and the visible region. You can use these lines to calibrate. They are basically called as the calibrating light sources. They can be used to calibrate your monochromators if you lose the calibration of your monochromators. Alternatively, you can think of using a laser also. You can think of using a laser like a helium neon laser 632 nanometer. You can put it in place of the light source and get it out of the excitation monochromator. Put a small reflecting mirror in the sample compartment, send it through your emission monochromator, see the output before the detector. That's the way one can detect the alignment or calibration of the monochromators. This is a way one can calibrate the monochromators also. So having learned the entire spectrofluorimeter, next step is what are the parameters that one can acquire from these fluorimeters. You can acquire the fluorescence spectrum, excitation spectrum, fluorescence quantum yield, fluorescence anisotropy, phosphorescence spectrum, and phosphorescence lifetime. To record the phosphorescence lifetime, you need a pulsing, another additional lamp is required, pulsing lamp, which can be used to measure the lifetime. Otherwise, with a normal spectrofluorimeter, one can measure all the 5 parameters. If you have an additional light source, which is a pulsing in nature, you can also measure the phosphorescence lifetime. These are the parameter, but the fluorescence lifetime cannot be acquired within a normal spectrofluorimeter. Now you have the fluorimeters combined with the TCS-PC also, which is a combination spectrometer, which is more expensive. But in a regular spectrofluorimeters, you cannot measure the fluorescence lifetime. But it is possible to measure the phosphorescence lifetime because it's a long lifetime, and compared to the fluorescence, what you need is a pulsing light source for that. Right. So as we already reviewed in the first session, what is an excitation spectrum? And just recall, you can record an excitation spectrum as I already told you. Here you have to fix the emission monochromator wavelength and then scan the excitation monochromator and lower than the emission monochromator wavelength. And then you same thing when you go for an emission spectrum recording. fixed in the more in the UV region or in the blue region of the excitation, and then start moving it away from the excitation monochromator, maybe 5-10 nanometer away from the excitation monochromator, and record your emission. This is how one can record an excitation spectrum and emission spectrum. We also discussed about the what is the difference between excitation and spectrum and the absorption spectrum. And you have seen a mirror image relationship between the absorption and excitation, absorption and the emission spectrum. Excitation and the emission absorption spectrum, they look similar, the other way they overlay each other, and not always. So you will also see a difference between excitation and the absorption spectrum when there is a geometry change in the excited state. If there is no change in the geometry of the excited state, and absorption spectrum and excitation spectrum will look the same. And in fact, excitation spectrum is used now. We also compare the absorption excitation. If there is no change in the excitation geometry, as I already told you, it will be the absorption, and excitation will be the same. In fact, excitation spectrum can be used to analyze the presence of multiple components in the medium. That is also possible when you record an excitation spectrum. Or geometry changes can also be identified. If there is no such system in your molecule, then absorption spectrum and excitation spectrum will be the same. You can also compare absorption and excitation. Absorption is a phenomenon from a ground electronic states, 0 vibrational state of the ground state to the excited state, vertically to higher levels of the excited states. It can be S1 state, higher vibrational states. Now relaxation, the excitation is basically from the 0 vibrational state of the first excited state to the different vibrational states of the ground state. That is how you can see the fundamental difference between absorption and excitation. And basically absorption talks about the ground state phenomenon, from the ground state to the excited state. Your excitation spectrum is from the excited state to the ground state. That is how you can try to understand the excited state properties by recording an excitation spectrum. And by recording an absorption spectrum, you can talk about the ground state properties. So this is how the absorption spectrum, excitation spectrum, emission spectrum can be compared, and you can also acquire this in a spectrofluorimeter. Now, you always come across some of the common problems when you record fluorescence. The one point is the concentrations of the solutions. And when you have the high concentration of the samples, then you have a problem of interference, and that is what is called as a inner filter effect. And if you have the solvent, the purity of the solvent also interferes with your measurements. And these are basically can be avoided. This pure solvent usage can avoid the contamination of the solvent. And you can see the second one is a scattering, which is a Raman scattering and the Rayleigh scattering. They can be really identified, and in fact when you record fluorescence, Raman lines interfere. It can be avoided by changing the excitation wavelengths. And you can also see the particles, floating particles in your sample. They also interfere, they scatter the light, so you have to filter in that case. So first point is inner filter effect, which can be avoided by diluting the samples. Always you take a dilute solutions. The second point is the contamination by the solvent is purify the solvent, and always use a spectral grade solvents. If you don't still have problem, and purify further and use it HPLC grade solvents. And if you are looking at the scattered light, and you can really change the excitation wavelengths, or use an appropriate filter, you can avoid all this scattering. And particles is remove the particles, filter the solutions and use it. This is how the common problems can be avoided. They can be overcome. Now this is to tell you about how exactly the sample issues the linear region of the fluorescence. When you take higher concentration, they are not linear. When you are looking at analytical measurements, always you have to use a linear region, as you are using in the absorption principles following Beer-Lambert law. You can also look at the linear region of the fluorescence. And if you are using an excess emission also, if you have a problem, emission is falling on the detector, then your emission also get distorted. Then you have to filter your emission intensities with the ND filters, or you should use a narrow slit pitch, or move away from your absorption peaks where the light is absorbed less intense. So this is how the PMT saturation can also be distorting your not only the emission spectrum, it will distort your emission spectrum. So that is the reason why you have to use a very dilute solutions and or use the filters properly, right? So this is the choice of the solution is very important. When you go for again a higher concentration, I am illustrating here how exactly transmission happens here. This is the highest concentration, this is the lowest concentration. You clearly see it in the highest concentration, and only in the very initial layers the sample is absorbing the light. Rest of the layer there is no transmission at all. So if you see that under these conditions, you may not be able to record any fluorescence at all, because the light is not really going up to the center of the cell where the emission is being monitored. Now you can see little more dilution. Maybe it's allowing, but it's not still not good. This is also a little better, but not perfect. Now the last one, it's completely allowing uniformly. Beam is there's no conical action of the light. You can see it completely uniformly passing to the solution, and you can nicely see that the fluorescence happening in this. So a very dilute solution is better. It will avoid inner filter effect. Low concentration is preferred. When you are looking at very high concentration, the inner filter effect, there are two types of inner filter effect. You will see it. Excitation path, which is called as the primary inner filter effect. You can also see the light that is emitted by the sample can be absorbed by the sample itself, which is the emission path. This is called as the secondary filter effect, secondary inner filter effect. So you should avoid both the inner filter effects by diluting the sample solution. So this is very important requirement. And you can see how exactly taking a higher concentration inner filter effect can really distort, shift the spectral positions. You absorption very dilute solution, you can see the dilute solutions and the higher concentration, so you can really see the shift in the emission maximum, also shape of the emission also changes. So this should be avoided. So again, I repeat again, very dilute solutions. Most important thing here is the preferred absorbance is 0.1. Absorbance should be lower than zero point 0.1, right? That is the most preferred solution concentrations. So where you are going to excite, absorbance should be somewhere around zero point 0.1 unit, and that will be very ideal for the recording of fluorescence spectrum. Now if you look at the sample cuvettes, different family of cuvettes, you will see different path length, and your 10 millimeter path length, 5 millimeter path length, and the materials are also different. Generally, you can use a quartz or plastic cuvettes or optical glass. So the quartz is the one used for the UV and visible region. Optical glass can be used only for the visible region. Plastic cells can be used only for visible region, and that is only for aqueous solutions. So the plastic cells normally can be use and throw type of a plastic cells, so they are very cheaper, doesn't cost much, so you don't have to have a problem of cleaning the cuvettes. So when you are using a quartz and optic glass, the important thing is cleaning is very important. If you don't clean properly, you will have a lot of contamination from the previous measurement. So it's good to have a very good cleaning of the cuvettes. The normal cleaning reagent that is being used is the HCl and alcoholic solution. That is the best cleaning solution for all the quartz and glass cuvettes. and you can use a lower path length or even a capillary cells, so low volume solutions. There are all different kind of measurements are possible. Smaller optical path length instead of 1 2 millimeter or 2 millimeter. So different types of the cuvettes are available for a different kind of a concentration of solution. Even if you have a low concentration long path length, high concentration small path length, all these can be used. Or even if you want to use a very high concentrated solution, front surface triangular cells, you can use it. You can see that excitation and emission here, so that is the advantage of using a front surface. You can also use a thin cells special compartments which can be used for the solid samples, also front surface materials. Right, when you go for a fluorescence, scattering is also a problem, and if you can summarize all the phenomena subsequent to excitation and can have a transmitted light, can have a Raman scattering, elastic scattering, fluorescence, and Raman scattering. Raman scattering use the anti-Stokes and Stokes lines, and they're all coming up from your excitation. So we are avoiding transmitted light because of 90 degrees geometry. When you are measuring at 90 degrees geometry, when you are measuring the fluorescence, the two scattering phenomenons will interfere. So we have to make sure the interference is not there when you're recording a fluorescence. These two are Raman and the Rayleigh scattering. Now, first of all, we should be clear how to understand or how to identify the Raman scattering, Raman lines. Otherwise, we know very well fluorescence emission is independent of wavelength of excitation. That is what is the Kasha's rule. Whereas Raman line shifts as you change the excitation wavelength. So you can see the relationship 1 by lambda Raman is equal to lambda Raman minus nu bar. Raman shift, which is around 3400 to 600 centimeter per watt, 3600 centimeter inverse for water. Now, as you shift your excitation wavelength and towards blue region, you can also see that the Raman lines also shifts towards blue region. Now you can really see this clearly, and different excitation wavelength. 460 excitation gives you Raman line at 546, 450 shifting towards little blue region gives you 532, little more shifting 528 and so on. So you keep shifting it, completely take away the Raman line out of this fluorescence. You can clearly see the only fluorescence. How exactly you can see it here, and this is as fluorescene with the PBS buffer, and you can see the Raman lines, and here you can subtract the Raman lines and get the true fluorescence also. So there are two ways in which you can remove the Raman line, one by subtracting from the full emission spectrum or by shifting the excitation wave. These are the two options available to avoid the Raman scattering in the fluorescence. Now, if you look at the Rayleigh scattering, now when you are exciting at 300, you will see the strong first order Rayleigh scattering, and also you will see at the 600 second order Rayleigh scattering. If you have Raman line, water for example 334 nanometer, it is 300 excitation will give you a 354. So if you have a sample emission here, you will have interference, and you also have some fluorescent contaminant, they will also interfere. So all these interferences should be removed. Fluorescence contaminants should be avoided by purifying the solvent and then better solutions, and Raman line can be avoided by shifting the excitation wavelength away from your fluorescence, and then your second order scattering can be suppressed by using an appropriate filter. This is how you have to make sure you are not getting an interference from the scattering and other things. Now here is a clarity that is brought in how artifacts induce second order scattering. This was illustrated in nanoscale because somebody has reported as if the second order scattering as a new emission. They observed it, and they have really taken the second order scattering. When you excited 200, second order scattering 400, and they have really clearly illustrated in this how to get the excitation at different wavelengths, different second order scattering, along with the sample, and how to avoid all that was really published very clearly. And they made a comment also in this nanoscale. And this is how, sometimes people confuse with the second order scattering as a new emission and publish it, but later on it will be criticized by the scientific community. This is the problem with most of the biologists, where they don't understand clearly about this instrumentation part, and that leads to some problem. And also this is the case with many of the material scientists. So the spectroscopist should have a clarity about this scattering phenomenon, and that should not be you know misunderstood as a new emission. So that is very important point here. Now, if you so far we have been talking about usage of a solution, recording of fluorescence in solution medium. Someone wants to make use of solid sample measurements. There are some solid sample holders available. They are rotating base, and this is the plate in which you can stuff the sample and then mount it and record it. Or you can also have a thin film samples also can be used here. Films can be used here. Now here is a geometry. You can see the excitation light and the emission light. When you are measuring at 90 degrees in this thin film and solid samples, you will also get a fluorescence and a specular reflection. So you have to, it's basically a scattering. So you should avoid this scattering light going into the detector. So rotate to an angle of another 30 degrees. So you will see it reflection, specular light will go like this, and fluorescence will come and all it will go to the detector. Fluorescence will always be there in all directions, whereas reflectance will go at 90 degrees. So that's the way you can avoid your reflecting light. And now I'm really illustrating here how exactly a rotation of 30 degrees can avoid the scattering. In scattering plus emission here. Now if I rotate 30 degrees, you can really look at the scatterings disappearing. So this is how you can make use of a solid sample holders also to measure the fluorescence. Now you also have another device called as the integrating spheres. Using the integrated spheres, you can record the fluorescence spectrum of a solid state. In addition to this, you can also calculate quantum efficiency of the solid material. So this gives a provision of a solid or any powder samples quantum efficiency. Now, there are other attachments which can really go for a variable temperature measurements, low temperature measurements. So you can have a thermostated cuvette holders. You can have a Peltier. Peltier technology is basically available, a little more expensive, and this is a water circulating type temperature variation. Here is a Peltier technology which can go from minus 40 to higher temperatures, whereas water is somewhere room temperature to higher temperature. And if you want to go for a liquid nitrogen temperatures, you can have a liquid nitrogen DVAR which can be used, and frozen solutions can be used to record the fluorescence. Or if you are using a phosphorescence, DVAR is a low temperature DVAR is important. So we will learn more about this when you are going for a phosphorescence measurements. So these are the temperature variable attachments are also possible with the spectrofluorimeter. So far we have looked at the basic components of the fluorimeter, and light sources, monochromators, sample compartments, and the detectors, and what are the problems that you face in the monochromator side and also on the sample side, and what are the interferences from Raman lines and Rayleigh scattering lines, and how do we go about measuring the solid materials and how to vary the temperatures in a spectrofluorimeter. These are the discussions we have made so far, and I'm sure this has given you a clarity about the component hardwares and capabilities of the instrument that are available. There are a number of manufacturers, they also make advanced machines. So these advanced machines can give you a synchronous scanning, and synchronous scanning is available in the normal routine machines also, but with you have an advanced machines which gives you an advanced feature called as a EEM, and here both of them will scan excitation and emission with the delta lambda. With delta lambda, zero can also be done, which can be used for the correcting of a fluorescence spectrum, which we will be learning it when you are measuring the quantum field, learning the quantum field component. Now here is a synchronous spectrum with the delta lambda can give you an excitation emission matrix. It's is basically a three-dimensional plot. One axis is the excitation, other is the emission, and third axis is the intensity. You can also represent this as a EEM matrix, which gives you the multiple presence of multiple components. So you can see that two spots are referring to the different excitation emission wavelength. This is a different excitation emission wavelength. The depth of the color is representing the intensity of the emission that you see. So this feature can be they're available in a mostly of advanced machines, which can help you to have the matrix of the excitation and emission, which can be used as an analytical tool to identify the mixture that are present. These kind of machines are manufactured by a number of companies worldwide: Job in Vion, Edinburgh, Hitachi, Agilent, Shimadzu, ISS, Jasco, and Berkeley. There are some other low-end machine companies are also available. So these are all the machines which has a lot of capabilities from spectral measurements, low temperature measurements, solid sample measurements, and also EEM measurements. I hope this has given you a complete overview of the entire fluorimeter, and go through this video. Enjoy it. All the best. Thank you.